Hi, hello to everybody. Thank you for joining us today. We will wait just 1 minute and then we will start so that anybody can connect. Okay, so just wait a few seconds. Okay, so let's start. Today we are talking about beyond ETO, mastering sterilization complexity in real medical device application. So the industry is moving past ethylene oxide, but how do you make safer alternatives actually work when they meet real-world medical devices? So today, Claudio and Lucia will explore real work case studies applying VH202 sterilization to diverse medical devices, and they will guide you through our proven operational framework, tackling process complexities and add-on. I will leave now the stage to Claudio and to Lucia, and thanks for your participation.
Thank you, Valentina. Good morning to everybody. I'm going to share the screen. 1 second. Okay, so good morning once again, and thank you for being here. Today we are going to explore the future of sterilization technologies, specifically why vaporized hydrogen peroxide are shaping up as the most reliable alternative to ethylene oxide. For decades, ETO has been the backbone of medical device sterilization, but things are changing and fast. And those changes have real consequences for manufacturers, regulators, and most importantly, for patients. Special attention is given to the challenge associated with the new modalities and how advanced techniques can address critical issues like residue management and sterilant penetration. Vaporized hydrogen peroxide has gained significant traction as viable low-temperature alternative for a broad range of sensitive devices, especially with its recent FDA Category A classification. Let me take a moment to present today's agenda. We will start with what sterilization by design means and why is the Fedegari identity. A quick overview of regulation and guidelines behind this transition from ETO to alternative sterilization method, a sterilization technologies comparison and materials compatibility to understand how the VHP sterilization process is able to meet your requirements, then H22 advantages and challenge, and how the deep vacuum up to 1 millibar is the solution to win this challenge. A deep diving in this sterilization process and especially on the equipment process and some case studies. Conclusion and QA section. Today, Fedegari is a global leader in providing highly customized integrated solution for the biotechnological and pharmaceutical industries, with several branch offices around the world, as we can see here on the right, in order to be as close as possible to our partners and customers. Sterilization by design is our identity. In our view, sterilization is not a regulatory checkbox or a process to be verified at the end of production. It's conceived and built upstream within the very design and management of the process. This is what sterilization by design truly means. Currently, about 50% of all medical devices worldwide are sterilized using ETO. So the reason is clear, it works. ETO is effective for devices that cannot tolerate heat or moisture, and it penetrates complex geometries extremely well. But that strength also comes with a serious challenge. ETO residue and emission are harmful to people and the environment. And that's why we are at a turning point. In 2019, the United States Environmental Protection Agency, EPA, identified ETO as a significant health concern, citing a cancer risk for workers and surrounding communities. This prompted the US FDA to call upon industry stakeholders to actively seek alternative sterilization methods. These concerns have already led to the closure of commercial ETO sterilization facilities. The collective stance of the EPA and FDA indicates that ETO is no longer considered a suitable sterilization method without significant warning. FDA has announced in January 2024 that it now considers vaporized hydrogen peroxide to be an established method of sterilization for medical devices, recognizing the modality's long history of safety and effectiveness. FDA's recognition of VHP as a Category A sterilization process, placing H2O on pair with moist heat, dry heat, ETO, and radiation. In terms of regulation, there are, as we can see here, the ISO 22441, which defines how to validate and control VHP cycle, and almost close to be released, EN 17180, which defines the technical and performance requirement for VHP sterilizer. Let's compare and focus on sterilization processes for heat-sensitive materials. So, as we can see here on the table, ETO is highly effective, but it leaves toxic residue, requires long aeration times, and face growing regulatory restrictions. H2O, on the other hand, decomposes into just water vapor and oxygen, leaving no harmful bioproducts. It's faster, cleaner, and more sustainable. The trade-off is that H2O penetration can be more challenging in complex geometries. But, as we will see on the next slides, with deep vacuum technology, we have been able to overcome those limitations. About the compatibility, here we can see a long list of materials. Each one has been tested in our laboratory test or is coming from literature. These materials are the common ones used for the manufacturing of medical devices like PE, PVC, PC, and for their coating like titanium nitride or DLC for pacemaker, for instance, or for surgical instruments, and of course metals like stainless steel for the filling parts or glass for the primary packaging. Advantages of VHP. For sure, low temperature processing, H2O can sterilize effectively at temperature as low as 40 degrees, with a typical operating range between 24 to 38 degrees. This significantly reduces thermal stress and potential damage to heat-sensitive materials that cannot tolerate traditional high-temperature sterilization methods. Broad spectrum effectiveness. H2O has high effectiveness against a wide range of microorganisms. When used for the sterilization process is consistently achieves a minimum of 10-6 of sterility assurance level. Environmental profile, green. H2O is considered a green sterilization modality because its active vapor breaks down into oxygen and water vapor. This eliminates the dangerous emission associated with ETO. Excellent material compatibility. As we have seen on the slide before, H22 has an excellent compatibility with a wide range of polymeric materials and metals. It's suitable for several products, including general surgical instruments, endoscopes, implants with electronics, pharmaceutical containers, and prefilled syringe. Regulation validation per ISO 22441, as we have seen on the slide before. FDA approval for healthcare sterilization of medical devices. So, despite its advantages, H22 also presents specific challenges that require careful consideration in process development. So, for sure, the first one is some material incompatibility. The primary challenge of H22 is its incompatibility with cellulosic materials. Cellulose degrades hydrogen peroxide, compromising sterilization effectiveness. Additionally, certain uncoated reactive metals, such as copper and brass, may react with H22, leading to surface degradation or discoloration. These issues need careful material selection for devices and critically for primary packaging. Second one, shape and complex geometries, and then direct contact needed, and some secondary packaging constraints. So to mitigate all these challenges of H22, particularly concerning penetration and residue management, deep vacuum processing is identified as a primary or preferred method to replace ETO and to improve the H22 penetration and the removal aeration. About the improved penetration, for complex devices, deep vacuum processing significantly increases the penetration by creating a pressure differential that facilitates the diffusion of hydrogen peroxide vapor into complex devices. Concerning to the efficient residue management, H22 offers a cleaner process compared to ETO, as its breakdown products are oxygen and water vapor. The application of a deep vacuum during or after H22 cycle further improves the removal of any residual vapor and its breakdown products, and decreasing or eliminating the need for extensive post-sterilization aeration, typically associated with ETO. As Fedegari, we are able to guide you in selecting the most appropriate sterilization pathway based on material compatibility, working alongside you in our tech centers to experimentally evaluate each specific application, developing and validating tailored H22 sterilization process for highly complex devices, ensuring safe and reliable outcomes. So here we see the main features of this process: a pressure up to 1 millibar if necessary, a temperature from 30 to 60 degrees, a tailored process based on product features with vacuum facilitating H22 penetration and improving aeration. Now the question is, why limit your process to one technology? As Fedegari, we combine two technologies in one equipment, moist heat and H22 deep vacuum. One machine, two suitable processes to treat and to sterilize the widest range of products and packages. Moist heat is applicable on glassware, solid loads, metal-like items like machine components, surgical instruments, tools, filters, textiles, stoppers in bags or no, wrapped items. H22 deep vacuum means medical grade disposables like tubing sets, blood lines, plastic or polymeric packaging, PFS, sealed or semi-sealed containers, complex shapes or heat sensitive materials. So I finished my first part and I'm going to hand over to Lucia.
Thank you. Thank you, Claudio. So, can you see my screen? Okay, so before moving to the experimental design, I want, and the case study, I want to briefly introduce the H2O2 sterilization process and its mechanism of action. So the process consists of three main stages: conditioning, preheating, sterilization, and aeration. So during the conditioning, the load is heated and the humidity is reduced in order to ensure consistent process conditioning. Conditioning is followed by sterilization phase, where the air is removed and hydrogen peroxide is distributed into the chamber in one or more pulses. And the load is exposed for a defined holding time. The sporicidal effect of hydrogen peroxide is based on oxidative damage to the cellular component, leading microorganism inactivation. And finally, during the aeration stage, the residual H2O2 is removed under controlled condition in order to reduce the concentration into the chamber and into the load below 1 ppm. At the Fedegari Tech Center, we develop sterilization process using a structured and data-driven approach. So the experimental design starts with defining the testing condition, including the chamber load configuration, the loading pattern, and the biological indicator placement. And after each sterilization cycle, the critical quality attributes such as the sterility, the residual H2O2 are evaluated, and based on this result, the critical process parameters are optimized until the required performance is achieved. The final cycle is then confirmed to demonstrate process robustness. So let's now move to the case study. So I will present three examples with increasing complexity to show the same approach can be applied to different products and challenges. So let's start with the first case study. The first case study is focused on a ready-to-use vial system with multiple sterile barrier. The key challenge here is H2O2 penetration across the different packaging layers. And to assess these, biological indicators were placed into a representative position within the system. The goal was to verify that the sterilant could effectively reach all the critical location. The process was optimized by adjusting the key parameters such as the vacuum level, the H2O2 injection pressure, the holding time, and the number of pulses. And this iterative approach allows us to progressively improve the process performances. As a result, we can summarize that both hydrogen peroxide consumption and overall the duration of the cycle, so the cycle time, were significantly reduced while maintaining the process robustness. Biological effectiveness was evaluated using a standard biological indicator for hydrogen peroxide, and as reported in the table, no growth was observed after incubation across all the tested locations. So these results indicate that the process conditions, so the critical process parameter, are effective in achieving the required sterility assurance level. In parallel, the residual amount of hydrogen peroxide was evaluated using two complementary analytical methods. For the cycle optimization, we use a kit. It's a colorimetric test method based on ferric thiocyanate. And we use this kit um, uh, as a screening kit in order to optimize the process. And then, in order to confirm the result in the final cycle, we use the UV visible method. And as reported in the table, for both methods, the residual hydrogen peroxide was below the limit of detection. This result confirmed that the sterilization performance, in this case, the parameter that we use for the aeration stage, are able to achieve and maintain control over residual levels. So, to summarize the study outcome, the sterility was achieved, demonstrating that the process and the critical process parameter selected are the proper one for this type of product. And then residual hydrogen peroxide remain below the detection limit and well within the acceptance criteria. And regarding the product integrity testing is currently ongoing with the preliminary result confirming no impact on product quality. So overall the data confirm the effectiveness and the safety of the sterilization process while maintaining a short cycle time and low hydrogen peroxide consumption. Let's move to the second case study. This case study is focused on the sterilization of pre-filled syringes containing thermosensitive ophthalmic drugs. This type of product is uh, could not terminally sterilize using moisture sterilization because it would compromise the drug stability. So at the same time, the sterility assurance remain a critical requirement for the product. For this reason, low temperature sterilization approaches need to be evaluated, ensuring both microbiological effectiveness and product compatibility. Okay, the process was developed through a stepwise approach. So we start with the small scale testing and we perform different sterilization cycle. We test different conditions. We change the number of H2O2 pulses and the total amount of H2O2 used for the sterilization. So the total amount of H2O2 injected during the sterilization stage. And based on these results, the optimized process is now being verified at a full scale with the final transfer to the customer equipped planned as the next step. This slide focuses on the key aspect of the H2O2 H2O2 process. So the sterilant penetration, and the aim is to verify that H2O2 can effectively reach all the critical area of the system, including the syringe barrel, the syringe component, the blister, and the thymic barrier. And understanding penetration behavior is essential because it directly affects the process robustness and the reproducibility. In particular, we need to ensure that all the relevant surfaces are exposed to the H2O2, even in the presence of packaging constraint and complex geometry. As for the previous case study, the process effectiveness was evaluated using biological indicator placed in the worst case position, and in parallel, the residual amount of H2O2 was assessed using both a screening and a confirmatory analytical method. And this approach ensures the evaluation of both sterilization performance and safety aspects. The results show that across all the tested cycle, the biological effectiveness was achieved for cycle number 2 and cycle number 3, with no growth observed after 7 days of incubation for the biological indicators. In parallel, as said before, the residual amount of H2O2 were evaluated, and as reported in the table for both the blister and the product drug, these values are below the quantification limits. These results indicate that the selected process condition allow to balance the sterilization performance and the H2O2 residual control. It should be noted that these findings are based on the current test configuration and should always be confirmed under the final process conditions. So, to summarize this case study, the sterilization process was successfully optimized to achieve the required sterility assurance level, and H2O2 proven to be suitable approach for the external sterilization of the syringe containing a thermosensitive drug. The residual amount of hydrogen peroxide remain within accepted limits based on the applied analytical methods. And overall, the result confirmed that H2O2 sterilization can represent an alternative to ethylene oxide, especially for heat-sensitive products where the traditional methods are not applicable. As always, the final process selection should be based on case-by-case assessment, including the product characteristic, the packaging configuration, and the regulatory expectations. So let's now move to the third case study, which focuses on the hemodialysis blood tubing set. This type of product represent a different level of complexity compared to the previous example, mainly due to the geometry and the internal pathways. As we will see, these characteristics introduce additional challenges in terms of sterile penetration and process control. So this tubing set present several features. So long tubing up to several meters, like 4 meters, and with a small internal diameter, and not all components are vented. So from a sterilization perspective, this means that the sterilant distribution becomes the main challenge, especially inside the small lumens. So to properly assess the process effectiveness, the biological indicator will placed in the worst case location where the penetration is expected to be most critical. And in parallel, the residual hydrogen peroxide levels were evaluated using an appropriate analytical method. This combined approach allow us to assess both the efficacy and the safety, particularly in a complex geometry. So the results. The results show that across all the tested location, biological effectiveness was achieved with no growth observed for the biological indicator after 7 days of incubation. In parallel, the residual amounts of H2O2 were consistently below 1 milligram per liter, and this result indicate that the selected process condition, the selected process parameter allow to balance the sterilization performance and the residual control. So, to summarize the outcome of this case study, the sterility was achieved across all the tested configuration, confirming that the selected process parameters are suitable even for a complex geometry such as long tubing set. And at the same time, the residual amount of H2O2 remain within acceptable limits, ensuring that safety-related requirements were met. The consumption are around 150 grams per cycle with a total cycle duration around 8 hours. So as we show, we apply a science-based framework where sterilization performance is always evaluated together with the material compatibility and the H2O2 residual control. And the aim is not only to achieve the sterility, but to ensure a balanced and a robust process outcome, supporting both the product safety and the product integrity.
Okay, so before closing, we would like to share with you what we just let me share the screen. Do you see the screen? Do you see the screen? Sorry. Okay, so before the conclusion, we would like to share with you, and here shown what we already test in our tech center, in our machine here in Italy, and then we can see let me say the easiest one, the easiest sample, which are the empty syringes. To the more challenged ones like hemodialysis tubing set and oxygenator. As we see, we test empty syringe, vitrectomy system, suture kit, PFS with ophthalmic drug, ready-to-use vials, pacemakers, filters, hemodialysis tubing sets, and oxygenator. Some of these are already ongoing, and quite soon we will have the final result. To close the webinar, for sure, we can say that increasing the regulatory pressure on ETO use demands the transition to alternative sterilization methods that are safe, effective, and compliant with global standards. So choosing the appropriate sterilization method requires a systematic assessment of product stability, material limitations, and regulatory requirements to maintain sterility assurance and product integrity. So vaporized hydrogen peroxide under deep vacuum is a reliable solution for complex and sensitive types of items, ensuring both microbiological effectiveness and material compatibility, offering an alternative solution to ethylene oxide sterilization. With its flexible process parameter like number of pulses, H2O2 amount, deep vacuum target, holding time, etc., the deep vacuum H2O2 autoclave allows adaptation to ensure process effectiveness and optimization of the H2O2 sterilization cycle. So I finish my part. We are here available for any question about it.
So thank you guys for this insightful presentation. We have received many questions and I would like to start from this one, which is related to one of Lucia's case study. That is related to the PFS case study. So for the prefilled syringe, how did you determine the sterilization settings for each test cycle?
Okay, so we use a biological indicator as shown in the picture, located in the worst case position according to the customer requirements, and we use a different number. We use different amounts, so with different number of pulses, in order to evaluate the capability of the sterilant to penetrate inside the blister and reach the biological indicator and kill microorganism. We use an overkill approach for this product, and we will test with the full load both the overkill approach and the reduced cycle time approach.
Thank you, Lucia. So we'll go to the next question, which I feel it is always for you. So is it possible to control the amount or concentration of the H2O2 during the cycle, and how to do that?
Okay, so we can control. So the answer to this question is yes, we can, because our machine could be equipped with a special tool in order to measure also during the vacuum the residual amount of hydrogen peroxide. This is important in order to better control the process and understand the amount of hydrogen peroxide that is present during each injection of the sterilant into the chamber.
Okay. So thank you very much. Next one is okay. So somebody is asking that is related to the latest part of Claudio's conclusions. Can you be more specific, Claudio, on secondary packaging constraints?
Yeah, so basically, secondary packaging constraints is related to carton box, typically in cellulose. So for sure, that is the most challenge, let me say, packaging that we are testing with our technology. But we are, we are is something ongoing, and quite soon we will be able to confirm, let me say, something new in the market. Just let's wait a couple of months, and we will come back to you with some interesting result.
Yeah, and one similar question to almost the same issue is about for commercial product sterilization. Can you process palletized product, or does it have to be broken down to fit into the chamber? So if you want to give some details about that.
No, for sure. And there is something that, for medical devices, especially, and to replace CTO 100%, we need to sterilize pallets, several pallets, which compose the load. This is something that for corrugate, as we saw, corrugate carton box, we can plastic box, we are able to sterilize that load. For secondary packaging in cellulose, we are in progress. Let's see on the next month. But for sure, let me say the site for this kind of technology is how many pallets we have to sterilize. And this is the reason because more and more we are getting requirements for really big autoclave. Here we talk about 20, 30, 40 cubic meters, means a certain number of pallets.
Huge number. Yeah. So another question is about the consumption. So what is the consumption of hydrogen peroxides?
Yeah, in terms of consumption, for sure, the proper and the exact value of ml of H2O is extremely related to the kind of load, the geometry, the complexity of the product, and the volume. But we develop a new VHP patent system, which is able to spread the H2O mixture inside the chamber to reduce the H2O consumption.
Let me add that the consumption is also related to the characteristic of the material. So if the material is a plastic material, we can have consumption that is higher compared to the Tyvek and glass material. So we need to evaluate every time the geometry, the material of the product, the packaging material, the number of pieces, so the amount that we have in our chamber in order to evaluate also the consumption.
Thank you very much again. So this question is about the VH2O2 as an alternative not only to ETO, but also to other possible sterilization methods. So the question is, do you think that your technology could offer an alternative to irradiation? I feel, but then Lucia can continue the reply, that I feel that especially for RTU, that can be an alternative. Is that right, Lucia?
So in this case, H2O2 sterilization could represent an alternative to other sterilization method. It is important every time to evaluate the property of the material that we want to sterilize in order to understand, first of all, the compatibility of H2O2 with the material, with the complex geometry, and with the characteristic overall of the product that we want to sterilize.
Thank you. I think we have just the time for the last two questions. One is about the type of hydrogen peroxide. So the question is, does your equipment need proprietary hydrogen peroxide?
Yeah, no, our equipment doesn't need any proprietary H2O2 liquid. Our machine can run with any kind of H22 on the market, coming from local providers, the most recognized international provider of H22. We can run with any kind of H22. We just ask a percentage of H22 around 35%. That's it. We can run with any kind of H22 liquid.
Great. So anybody can buy the H2O2 on their market according to the place where it is more convenient for them. So last
Yes.
question. Can you provide a list of your facilities that perform R&D process development?
Regarding the H2O2 sterilization, we can perform process development just here in our tech center in Italy, in Albuzzano, very close to Milan. Then we have other 3 technology center, one in US, one in Brussels, and one in Singapore. And in this tech center, we can offer the process development for washing, for moisture sterilization, both by direct contact and for closed containers, so air over steam and water cascades, and other process such as the decontamination using H2O2. But for sterilization with H2O2 only here in Italy for now.
Yes, Lucia, you're right. So generally speaking, our tech center can perform different processes in order to develop cycle, optimize cycle, and so on and so on. For the time being, for H2O2, we are concentrating the test here in Italy, but also in order to concentrate the results as well in one only place and can make a comparison test comparable with different material and create a kind of library of experiences. But of course, in US, we are also starting this activity as well. So I think that time has run as usual. Thank you, everybody, for participating to this webinar. The video recording of this webinar will be sent to all participants, together with the replies to all the questions that we have not been able to address during this session. So thank you again, and we hope to see you at our next webinar.
Thank you.
Thank you.
Bye-bye.
Bye. Bye.